It has long been known that the rate of healing of differently shaped skin cuts caused by surgical operations or accidents varies. However, there is little research on the cellular mechanisms that cause this variability. A series of experiments conducted by researchers from Nanyang Technological University in Singapore in recent months have revealed that the shape of the cut affects cell movement. According to the study, cells whose movement is relatively less restricted in curved or wave-shaped cuts accelerate the wound healing process. Embryo development, tissue repair, and wound healing all depend on the ability of epithelial cells lining the outer and inner surfaces of our bodies to close gaps. In the experiments, the researchers used the MDCK cell line, which is often preferred in biomedical studies because it exhibits similar properties to epithelial cells in human skin. They examined how these cells closed the cuts made in an experimental medium consisting of hydrogel and mimicking human skin. In the experimental tissues, straight and wavy cuts were made with widths ranging from 30 to 100 micrometers. Cuts with radii of curvature of 50, 75, and 100 micrometers were also created to mimic narrow and wide wave-shaped wounds. The researchers used an optical measurement method called particle tracking velocimetry to observe the mobility of MDCK cells as they closed the wound gaps. The researchers found that the cells followed vortex-like paths in the wave-shaped incision, while in flat wounds they moved parallel to the edges of the incision. The cells in wavy wounds, which were given more room to move, were observed to rapidly bridge between the two sides of the cut during the healing process. This bridging of epithelial cells to close the gap in the skin is called reepithelialization. After 64 hours of observations, it was found that the healing efficiency of wavy cuts was about 5 times faster than straight cuts. According to the researchers, this difference occurs because cells in wavy cuts are exposed to more mechanical stress. However, they also observed that the flatness of the wavy cuts, the radius of curvature, did not make a significant difference to the rate of healing. The width of the cut had to be narrow enough to allow bridging. The experiments showed that epithelial cells were unable to bridge wavy cuts wider than 75 micrometers. Cells in areas with straight cuts, on the other hand, failed to bridge even in the narrowest wounds of 30 micrometers during the observation period.